Hello, everybody. I'm being told that we are live, and as I always do, I'm just going to wait a second to make sure everything seems to be working well on YouTube's end. And once I see that it is, I will actually start. And we're working. We're good. Great. Okay, so it looks like everything's good there. In the chat, just let me know if the sound's coming through okay. See a couple people here. We got a couple people watching. Great. I'm just going to wait to see that I can audio and video good. That's what I like to hear. Thank you, David P. Neff. So, hi, everybody. This was a little bit short notice, but Sal and I have been trying to get together to chat for a couple of weeks. And, it's like, hey, are you free this evening? And we're both free this evening. So my guest tonight is Sal Cordova, who is... Right. Yeah, I got the orientation right this time. Sal Cordova, right here. Many of you may know Sal. Sal, I'll let you introduce yourself because I don't want to butcher it, but I know there's a couple different titles, research assistant. So I will let you introduce yourself briefly, and then we can then I'll kind of get into what we might talk about. We'll see. We'll see what we talk about.
I'd like to thank Dr. Dan by mutual agreement. He said I could rebroadcast this on my channel. So greetings to all my viewers on my channel. And I'm Salvador Cordova. I'm a molecular biophysics research assistant. I do not fancy myself as a scientist in this area because I'd be ashamed if some of my colleagues, I would, compared to some of my colleagues, I'm just a data clerk. And so I emphasize that I'm a research assistant in these areas. I did hold the title of junior scientist when I was in the aerospace and defense industry before I got promoted to senior engineer. So that's just a little background. So I'm still under contract with Dr. John Sanford. It was supposed to wind down this year, but it's still continuing. And so that's a lot of fun. He and I mostly agree on things. You may hear me say a few things where I may differ from some of the things that he's written. And, he didn't hire me to be a yes man to prove everything that he articulates. He hired me because of my math and computational skills. And he wanted someone that would give him information that he was seeking. And, I've had the privilege of working with him for 7 years. I have a YouTube channel. I actually have two YouTube channels. One is Evidence and Reasons. That's a short title for Evidence and Reasons for the Christian Faith. And then another one, Evidence and Reasons Academy, where I just, at this point, it's a small channel, but I'm trying to do lectures on chemistry and basic science, and in part thanks to Dr. Dan here, who got me started on the OpenStax material. So both Dr. Dan and I are trying to build out our channels. We can't monetize it yet. So even though people have offered to give us super chats and whatever, thank you for the thoughts, but we can't do that right now.
Nope. If you're watching Sal's channel, sub to me. If you're watching my channel, sub to him for just, cross-pollinate. All right.
Yes. So if you all see me looking here, I have a second monitor, and I will try to, I will look at some of the chats. I'd like to thank David Neff for affirming that the audio and video are working well. So greetings, David.
So, and I see Leophilius put Evidence and Reasons in the chat there, so we got a direct link to Sal's channel. Great.
Thank you.
One of the things, kind of how this came about, is we talked on Sal's channel a little while back about some of Nathaniel Jeanson's work, but last summer actually, Sal and I had a couple of debates on genetic entropy, and in one of them, they were on modern day debate, and in one of them, we really started going down a rabbit hole on ENCODE and junkDNA and methylation sites and what's functional and what isn't. And at some point midway through that conversation, we both realized that we were putting poor James's audience to sleep. So we kind of brought it back a little bit to the topic at hand. But since then, I've been really wanting to really dig into the nuts and bolts of that. And Sal is a great person to do that with. So I guess, Sal, if that's okay with you, I think that's a really fun thing to talk about. I don't know how many other people in the world think that's a super fun thing to talk about, but I think actually that was part of my professional job. I was sent
Dr. Sanford sent me to the NIH. He said, that seems really a big deal. Just find out what's going on. So I had the privilege of having NIH professors. They have us, I don't know how many people know of Cy Gart. He was actually professor at the school I was going to at the NIH. The NIH has a small graduate school.
Cy Gart.
Oh, what's that?
That is his book.
Oh, Cy Gart. Okay, great. So the NIH has a small graduate school. It's unaccredited, but it's kind of funny to say that because the PhD students at Johns Hopkins will get credit there, PhD credits. A lot of my classmates are postdoctoral students or PhD students. So it's just really funny. It's an unaccredited school, but the students are really very accomplished. And anyway, I was sent there, and my mission was just to learn what was going on and interact with the researchers. And so probably tonight I'll report on their perspective. Some of it is intuitive. There are probably some published things that have been controversial, like the claim of 80% functionality. I will say this, that
a little bit, yeah, a little bit controversial.
Sentiment, just the pure sentiment there, because they are medical researchers, is that it's there for a reason. If it's in the genome, it's there for a reason. That's just their, I haven't run into a researcher that didn't feel that way. So just qualitatively. So tonight we'll probably talk a little bit about that. I will say up front a little bit about my history. I accepted evolutionary theory. I'm a creationist now, a young earth, young cosmos creationist. I accepted evolutionary theory growing up because that's what I was taught in public school. I was raised in a Roman Catholic home that didn't care about these things. So I come from that background. I was an old earth creationist most of my life. I became probably an old earth creationist around, say, 16. I don't remember. That was a long time ago. And for most of my life, I was an old earth creationist, then a young life creationist, then young earth creationist. So why did I bring that up? I don't know. Just wanted to set the record straight on how all this came about. And I should mention this. We had an informal poll at my channel among creationist students. I said, who would you rather have as your teacher in biology? Would you rather have Dr. Dan, Kent Hovind, or P.Z. Myers? Unanimously, we voted for Dr. Dan. Now, that offended some of my creationist colleagues. They couldn't believe we said that. I said, if you're going to graduate school, if you're going to study biology, you'll understand why we picked Dr. Dan and not Kent Hovind. Sorry to say that, guys. I know.
Hey, Sal, just so you know, are you doing a simulcast right now, or are you just going to put this up? Okay, cool. You're just going to put it up. Okay, because somebody speed checked, and it's just making sure.
No, I gave up on that. It was just blowing up my computers. Couldn't handle it. Either it's my computers or the internet connection. It just couldn't handle that much.
Cool. So, just making sure.
Yeah. So Dr. Dan, okay, you're respected among creationist students that are actually science students. Okay.
I appreciate that. That's nice to hear. So with so just a little bit of history for everybody with the saga of junkDNA and ENCODE. The big splash was in 2012 with ENCODE's original finding, and I'll just put it up right here. It's one of these papers. Here we go. This is the one. It is called an integrated encyclopedia of DNA elements in the human genome. And I will show this because, why not? We can share screens. Here we go. Yes, the tips. This one. Here you go. So here's this. Oh, I should probably add it to the stream, right? I'm good at this. I know what I'm doing, everybody. Here we go. Okay. Integrated encyclopedia of DNA elements in the human genome. The big thing here, the kind of takeaway headline was, these data enable us to assign biochemical functions for 80% of the genome. Like, and I'll just zoom in on that. Like, that's it. That is the thing that everybody took away from this, is that, 80% of the genome is functional. Wow, that's a big number. We didn't think it was that high, because for reference, only about 1.5% is like exons in genes that actually code for an amino acid sequence, ultimately for a polypeptide. So 80% is a lot. And they go through in quite a lot of detail the biochemical evidence for providing this 80% number. Just looking for some nice figures here. Yeah, so like they have a lot. This is one of those big papers that has tons of really pretty figures, and they talk about things like protein binding of DNA. They talk about things like transcription, and they go through and kind of assign, they show how you arrive at that 80% number. And creationists took this and ran with it because intelligent design, right? You say, whoa, if most of the genome was functional, that looks a lot more designed than a genome that's maybe 5 or 10 or 12 or 20% functional. So that's kind of the original big splash, and there has been robust discussion of this topic since this paper was originally published in 2012. And one of the biggest kind of things was actually Dan Grauer, who, I don't know if people are familiar with Dan Grauer, but biologist Dan Grauer. He gave a talk in Chicago at a conference in 2013, and he said that if ENCODE is right, if the genome is actually 80% functional, then evolution is wrong. Now, that became shorthand for ENCODE disproves evolution in creationist circles. What he actually said was a lot more nuanced, and I know that because I was actually at that talk as a graduate student, which is really cool. I was actually there, and the point was not that if ENCODE is right, then all of evolution is invalid. What he was saying is we need to rethink things like the relative importance of strength of selection versus neutral evolution, for example. So that's what he meant by it. But we kind of ran with that one. And then ENCODE did a kind of a revision. They've continued to update their work. 2014, they published a major paper kind of updating their findings, where they significantly scaled back their estimates of functionality. And now it's one of those things that just kind of simmers in the background of evolution and creation discussions that, well, does ENCODE actually show that the genome is 80% functional? If it does, what are the implications of that? Sal, what would you like to add, if anything?
Could I share my screen?
No. Here, let me remove mine. And we'll add yours.
This is from just to give a little context here, this is a 2021 paper. I mean, this is like really March 10th, 2020. I'm sorry. And when they came out with that ENCODE thing, even at that point, even though I'm a creationist, I was just like, guys, just calm it down. This is really premature. And I've said no one knows how much is functional, and we don't even have great definitions of functional yet. Just, I just wanted to say all sides, we just need to temper something here. On the other hand, I sympathize with the medical researchers. I was at the ENCODE 2015 users conference. I got to meet the authors of that paper, some of them, the lead authors like Mike Pazin. And that ENCODE paper had like 160 authors. It's insane.
It was a lot. That's a lot of authors.
I mean, this was like all these institutions. And
it's so many they didn't even list them. They just said the ENCODE consortium and just left it at that because there were too many to actually put on the paper.
And the reason this is so large is this was a $500 million investment. It's just gigantic. And so this is their rollout of it. Now, their goal was not to prove functionality or non-functionality. Their goal, if I could summarize it, there's a paper co-authored by Francis Collins, the head of the NIH, that 90% of heritable diseases are associated with the non-coding regions. Correlation does not imply causation, but that was kind of the sentiment. So as I was talking to all the researchers, they said, yeah, we're dealing with this disease, and we find changes in the, we find an anomalous looking DNA, and it's, by the way, in the non-coding regions. So this is where that sentiment came from. One is right to be skeptical how well that sentiment can be extrapolated, but this is where it developed. It wasn't like they had some conspiracy to try to prove design or anything. This is where so I'm just putting that out there. The reason that I showed this later paper, I said this was more of what the substance of what would be needed to maybe help settle this argument, because when they're saying it was just transcribing and doing this, it's like, well, that's all well and fine, but it could still be junk. These could be RNA junk transcripts. So when I heard creationists quoting this, I just kind of cringed. I said, you still don't know. You still don't know just because it's being transcribed differentially according to cell type. I mean, the intuition is yes, but it's like until you start to elucidate the actual biochemical and physical interactions, it's still just kind of intuition. And I said, I'm just not going to go down there. As a creationist, I've said, I'm perfectly fine arguing that maybe a lot of our genome is disabled and no longer functional. It may have been created. We just don't, the way to settle this properly is as if we move forward in just our experimental and observational understanding. I certainly have my intuitions, and I'll say this is what makes me believe that it's functional, but to be ruling absolutely that it's a done deal, I feel uncomfortable with that. And so that's probably what gets me in trouble with some creationists. I will try to say, hey guys, I'm with you in spirit, but if we're teaching this to the next generation, I think you need to temper your words a little bit more. This is just not healthy to be taking shortcuts. And that's kind of my feeling. So that being said, I did show something there with the L1s and signs. These are large segments of repetitive elements. And this is a great paper. If I may actually do a journal club reading of this paper, because this wasn't around when ENCODE published its paper way back in 2012. They've had a lot of follow-on projects, hundreds of millions of dollars being invested that followed on. And I'm a little disappointed that some creationists still keep coding ENCODE. It's taken a while for me to persuade them, start saying 4D nucleome and then these other things.
Or at the very least, if you're going to quote ENCODE, don't quote the 2012 paper, because like that same group of authors has had a bunch of follow-ups that have actually gone into a lot more detail. The big one was the 2012. But I also, before I go any further, I want to say, Sal, I really appreciate that approach, that don't want to get over your skis approach to this, which is a very I think it's indicative of someone who's actually been in a lab and has done this, when you have to be like tentative. And if you get up in front of a lab group and be like, here's my data, here's what I did, and here are the big glorious conclusions, and then if you can't back it up, then you have a problem. And in bringing that approach to evolution versus creationism, I think is warranted. But let me just show a quick thing here, because I want to show two figures from the 2014 paper for everybody, because this paper really, I think they do a really nice job of, in a 30,000-foot way, breaking down the categories that they looked at and exactly why the original definition that they used was biochemical activity. And that could have been anything from transcription to being a methylation target to being a protein binding target, any of that stuff. If you had any one of some very broad set of activities, in 2012, that was lumped into that 80% of possibly functional, right, that they called biochemical functionality. And then in this 2014 paper, which they actually listed the authors here, and I think this is not all the authors, but this is the main authors, all of whom were on the earlier paper. This is defining functional DNA elements. And first thing is down here, figure 1, which I really like because, well, I don't like it because it uses area to delineate magnitude, and the human brain is really bad at determining surface area for circles. But putting that aside, taking your whole genome as just the circle, and then the shading in blue by level of activity, I think that's really nice. And then the protein coding, tiny little fraction, conservation, I think is really useful. And then phenotype associated. So that stuff like Sal talked about, where maybe you have a non-coding region, maybe it's just repeats, but if you mess with it, that leads to maybe a disease, right? So there's a phenotype associated with it. And a lot of this stuff overlaps. So you've got, for example, a very large fraction of the protein coding region is also conserved and associated with the phenotype. And that makes perfect sense. That's exactly what we would expect, right? And also has like high levels of biochemical activity, exactly what we would expect. But then you can zoom in on these data a bit more, and that takes us down to figure 2 down here, which is the really important one, because this is where it shows the relative levels of activity for a bunch of different classes of stuff in the genome. So you've got RNA right here. So this is polyadenylated RNA is this column, non-polyadenylated RNA, all RNA right here. And for each of these different classes, you have different scales, and we do not need to go into what these numbers mean. It doesn't matter for interpreting this. What matters is that, one, you max out at around 80%, and its transcription is the most frequent thing that happens, which is weird. You might think it's like some specific kind of DNA binding, but transcription is that, like, all RNA is actually the most it's the modal thing that goes on. But then the scale here, if you read through the paper, anything about 0.5 or lower, so the white and then the 2 shades of yellow here, that's indicative for a region that's transcribed into RNA of fewer than 1 transcript per cell on average. And like, if you're only getting 1 transcript per cell, that's not doing anything. The half-life of RNA, especially if it's not polyadenylated, is like that. Like, Sal, you probably know better than me what the half-life of RNA is. Like mRNA, it's in minutes. Do you happen to know, like, with a little precision?
No, your figures, or I'd actually say your figures are better than mine. I will point out, it is, as far as I could tell, some of this is cell-specific. That
yes,
the expression will be significantly stronger in some cell types than others.
Definitely.
I don't know. I don't know if this graph is reflecting that.
Yeah, I don't think this breaks it down by, I think some of these down here along the x-axis, some of these are, I believe, different cell types, I think. I'd have to go back and read it to be sure. But so I think we have there is a little bit of that specificity shown here, but obviously for the RNA, they just collapse it into 2 classes, and they're not showing like a lot of different specifics in terms of which cells are doing what.
So, I mean, I would just say if we see just a huge change depending on one particular cell type, then at that point, I would say I wouldn't write it off as being insignificant, because it could be very significant for that cell type.
That's definitely true. Yeah. But yeah, so going back to the RNA here, so anything in these yellow regions is going to be, that's like very clearly spurious transmission. And I think they actually say that somewhere up here. They go through the evolution, somewhere in here they actually say, like, yeah, at these thresholds, we basically say this is not like a functional RNA. It's due to just accidental transcription, essentially. And they actually go into, to their credit, they actually, in the 2012 paper, they didn't get into kind of the evolutionary biology of it. They were strictly biochemical. In this one, they actually do get into the evolutionary side of things, and they say, look, we would not expect all transcription to be like 100% of transcripts are functional and there is 0 spurious transmission, transcription. The energy and the precision required to do that would not be beneficial over long evolutionary timescales. You're going to have some wiggle in those regulatory systems. So we would expect whatever fraction of this to be incidental, and they draw the cutoff at about between 0.5 and 1 here. So you can see that you take that 80% transcribed number that like really grabbed everybody's attention from that original study, and that drops down to somewhere in the neighborhood of 30% once you account for the differences in the amount of RNA. And that's considering different cell types. So if it reaches whatever threshold in at least one cell type, it gets in that category. So I think this is really strong evidence that most of that transcription that's going on is not selected to do something. It's just incidental to whatever's going on. Some of it's going to be, you know, adjacent to a transcription start site. It's a little bit leaky. Some of it's going to be transposable elements or viral remnants that aren't entirely shut down, and they're doing a little bit of transcription. I think there's a lot of classes of things that we know of that are going to fall into those low levels of transcription category that aren't contributing to the physiology of the cell. Does that sound about right?
Yes, those are some of my sentiments. That's why I said I was trying to caution my colleagues. It's like, you know, this is this is too early in the game to be trying to proclaim victory. And what has disturbed me is that, it's part of the creationist mindset to be trying to convert people. Just like, guys, if your priority is evangelism and trying to persuade people rather than, thinking like a scientist, this is what ends up happening is you just overblow things. And, professionally, and also someone that's trying to teach other creationists, particularly science students going through school, it's like, I just try to say, you got to dial it back. This isn't healthy. I'm with you believing it's functional, and one day I believe we'll find out. We'll find out, and we keep finding out, and that's just what I've said. So everything you've highlighted has been my major concern, and I saw that right away. I said that's not a great definition, just because it's being transcribed. And just like, well, as I'm just repeating myself, that's not proof of anything. I mean, it may give you some hints and some clues, but until you know what those RNAs are doing, it's a little bit premature. So fast forward to this 2020 paper, I did not see that coming, where the RNAs from the L1s and B1s are being transcribed. They're actually connecting right back to the DNA, and it's part of the regulatory complex. Now, I actually had, in preparation for our debate last year, I was going to put that on the table, and I never even bothered pursuing it further because I had so many other things on my plate. I'm in protein biology, not genetics. So, and then I got recruited to do other things. I'm just like, goodness, so I had to put that on the shelf. So, and then now this March paper that was also talking about how the let me just share the title and
go for it. While you're pulling that up, just refresh my memory. So L1, are those signs or lines?
They're lines. That's the biggest. That's the biggest class. That's about 17% of the genome.
That's a big chunk, and those are derived from. So for everyone who isn't super immersed in this, lines, it's long interspersed elements,
and they're nuclear elements, I think.
Oh, nuclear, yeah, right.
Short interspersed.
And signs of the short.
Distinctions are kind of unfortunate because they're they just throw a number, just like
what's long and short? It's 3 nucleotides is short, but like how long does it have to be before it's long repeats?
Yeah, it's like that's just such an arbitrary to say long intergenic you think it has one function and it doesn't. I mean, right. It's almost and so I'm just throwing this out there. I said, this is more of what I said, this is what I was trying to tell creationists. Just wait. I know you're eager to evangelize and try to prove your case. I said, I've been in this for 45 years, starting with me being in junior high, being taught the Urey-Miller experiment. I mean, that tells you how old I am. I am getting to be.
I learned that one in, I think it's high school. I learned that one for the first time. But here, actually, there's an interesting discussion going on in the chat right now about what is variously called the C-value paradox or the onion problem, depending on who you talk to. Are you familiar with that one?
Oh, yes. Yes. And so what I have said is,
hang on. Let me just introduce it for everyone so we're all on the same page. So people are asking about, is there a correlation between the percent of the genome that we can document as functional, or broadening that question out, the number of genes or the size of the genome itself, and the complexity of the organism? And oddly enough, the answer is no, not really.
Like, so you and I are in agreement. No, it's what they call the G-value paradox. Yeah,
right. And it's weird because it's not even like, so the largest known genome is an amoeba, amoeba dubia or something. I forget what entomia. I don't remember what family it's in or what genus it's in, but some kind of amoeba. And it's got a genome that's like 500 billion base pairs. It's enormous. And then,
just as a caution, I think they overstated that number. It's been revised downward, but that still doesn't negate the point that you and I agree.
Right. It's
not a good measure.
It's still in the hundreds of billions, though. And it's massively polyploid. So it's not that it's 100 million, distinct things. It's polyploid. It's huge, right? But the you can even see variation like that within families. And the famous example, and the reason it's called the onion problem, is that onions have a genome that's about 30 billion base pairs. And it's a haploid genome of about 30 billion base pairs compared to about 3 billion for humans. But within the family of plants, you get enormous variation in there. So you get some plants in that family that are in the single digits billions. You get others that are in the 10 to 20 range. You get some that are above onions in the 30 range. And the question for creationists is saying, okay, if all that stuff is functional, why does the onion, or if most of that stuff is functional, why does the onion have 30 billion or whatever it is? And then this very closely related plant only has 5 billion. And what's going on there that explains that deviation if most of that stuff is functional? That's framing the question for everybody. So Sal, take it away.
Perfect thing, legitimate question, and I think the issue is the sensitivity to variation. I think these creatures with huge genomes, the variation in their DNA, let's say they have random mutation, it may be a little different than the issue with the humans. And we barely understand the human genome and all that. I mean, we're only just like this paper I was trying to share. It's just showing how little we know. And I actually think that each major species uses the genome in different ways. And we won't know until we just actually explore more, because these are such open questions. The medical community, just in general, the medical research community does believe that there's substantial sensitivity to variation in some of these parts, that you can mutate a little bit, but there comes a point, enough point mutation is going to be bad. Definitely translocations, frame shifts can be, I mean, in the coding regions, they'll be devastating, but we know that translocations, now that we know more about the 4D nucleome, we could have, we have maybe 200-300 canonical cell types, but other researchers will say, well, that's kind of an arbitrary classification. There could be thousands of cell types. The plants don't have as many cell types. And so that this whole thing of how the repetitive elements are used in humans could be very different than the way it's used in other creatures. The amoeba, I believe, would be a single cell. Mm-hmm. It's a different ball game with human beings where it could be much more sensitive. And I had a abstract peer reviewed in the FASEB journal. I'm not good. I'm not senior enough to have lead author status. So anything that I do peer review, this could be a big year for peer reviewed publications. I have to grab someone else's coattails to be able to be a co-author. But one of them dealt with topoisomerase, and I was arguing that it looks like humans are going to be a lot more sensitive to variation, definitely in the protein level. I would think the same for the non-coding. So if I could show that, this one here, this article.
Yeah. Oh, yeah. Let me pull it up. It's the one you have shared?
Yes. And this is a big enough deal that I think I might do a journal club report on my channel. It's genomic repeats, categorize genes with distinct functions for orchestrated regulation. So we have these L1 repeats, and it's just like, yeah, I mean, I can understand that people would think that this is just junk, because it's just like, why do you need a million copies of a phone book? You only need one. And so understandably, people said, well, why do you need all these repeats? And so this paper explores it in terms not as a coding thing, but as actually a physical substrate. And by the way, it pains me to say this, DNA is not a code. It is.
Normally in this conversation, that's going the other way, because I just spent so much time talking to Maddox about this, and we agreed it was a code.
I'd say, well, DNA is a substrate that can be used to implement a coding system. But one reason I threw that out, this is an example of DNA being used as a structural edifice. And so when we had all these expressions of repetitive elements in creating these RNAs, apparently the RNA, see, it says here, the RNA, a key role of L1 repeat RNA in sequestering L1 rich sequences. So there's some sort of binding here, and they have these really cool graphs where they show.
I just want to put a pin in that, and I want to go back to that figure in when you're done, because I think that figure legend says exactly the reason that I think that's evidence that most of that stuff is non-functional.
Okay, and that's really all I had. I think, you know, we have authors like this saying that they think that the L1 has function. So I'm not going to rule one way.
Can you put that back up though?
Oh, sure. So my point is, this is definitely not the argument that ENCODE was making.
Right. I mean, this is now 8 years later. We have much more sophisticated arguments going into this. Yeah, here. Let me pull that back up. Here you go.
So you wanted me to go into the legend here? Yeah,
that same figure you were just looking at. Yeah, here we go. So this is actually a really good indicate. Like, this is a great example of exactly where we disagree on the really minutia of this stuff. So what this says is striking association, genomic repeats, gene regulation, demonstrating a key role of L1 repeat RNA in sequestering L1 rich sequences and associated genes in inactive domains for silencing. And that's the key right there. Because what this is saying is that the transcripts from these L1 regions are involved in suppressing further activity in those regions. And I am not at all surprised that you take a region that is tightly regulated and suppressed, and if you mess with it, then that causes problems. That doesn't surprise me at all. If you start even just putting aside whatever region it is that is now being transcribed that shouldn't be, that's just going to gunk up whatever works are supposed to be going on in that cell. And I think you could point to those regions and say the fact that you have a phenotype, like a problem associated with changes to those regions that prevent that association, that silencing. Not only do I not think that's evidence of function, because the biochemical thing that's happening there is shutting that sequence down, I think that's evidence that that region is non-functional and specifically could potentially be harmful. And that makes sense in the context of most of these things originating from transposable elements, whether it's retrotransposons or DNA transposons or endogenous retroviruses, right? You've got parasitic genomic elements that get in there. And we're using various means to say, okay, you're stuck in the genome. I can't get rid of you, but you're going to stay there and you are not going to be expressed. You are going to sit in that corner with your methyl groups and your RNA and your histones, and you're not going to do anything. And yeah, go ahead.
If I could share this other one here.
Yeah, absolutely. Here you go.
This is the companion paper. This one talked about the roles of line 1 in mammalian development. And there are times when it's shutting off and then expressing. This one was actually also talking about the line 1's being used for organization. So probably, you know, I think I'm going to try to take my own advice here and say the same thing to do is just to go through these papers and just read them, because that would be doing the topic justice. But this paper was actually the one I wanted to talk about in our debate, and we stopped because we made an agreement on time limits. And I said, you know, I'm going to respect that. I'm going to honor that. So this one talked about essential functions of line 1 RNA in regulating both RNA and 2-cell specific gene expression. So this one did stand out to me. And there are other functions of line 1. And so this was from bioessays. This is also 2020. So these are discussions that need to happen. And I think the takeaway is...
would you mind putting that back up, actually?
Oh, sure. The takeaway is, you know, for my creationist colleagues, I'm on your side, and I believe this, but I just want to say, you know, maybe a little patience is in order, and to be just a little bit more cautious in your choice of words. 'Cause I know you're really eager to convert people, but this, to be trying to weigh into this, this is, if you just read through these papers, you'll see this is non-trivial stuff to figure out what's being said. And if I'm having a hard time uncoiling what it's saying, then maybe all of us just need a little bit of just sit back a little bit and say, okay, let's, just wait till the smoke clears and we have a little more understanding of what's going on. So this, I don't know how I stumbled on these things. I think what had happened was I was seeing stuff in bioRxiv that was talking about line 1s and B1s important for the 4D architecture. And so this was related to creating the 4D nucleome. So go on.
So this paper is an example of the same kind of thing as the last one, though, because in that figure you showed where it had the cell and then it had the figure legend under it that spanned the page, in that figure legend, it said this is specifically early in embryonic development, and it's in the pre-implantation phase. If you don't mind scrolling down to that, just to that figure, just so I can see the exact wording. So I want to make sure I have it right. It was that one. If you look at the figure legend there. So we're looking at a pre-implantation embryo. So first thing is that this is super, super early in development. And a couple things I want to point out here. This is 0 to 6 days post-fertilization, basically. This is basically from fertilization to about 5-6 days after is when implantation is going to occur. And first thing is that this embryo, this structure, has the cells are indeterminate at this stage. So none of the regulation that's happening at this stage is set in stone. These are all, until gastrulation starts, till some folding starts, these are all totipotent stem cells. These are completely you could remove some of the cells and they'll just grow more. They don't care. You can do whatever you want as long as you don't destroy the outer layer and the inner cell mass. As long as there's enough cells at this stage, it doesn't matter what these cells are doing yet at this stage. And I shouldn't say it that way. It matters. What I mean is they're not determined yet. So any gene regulation, any on and off, is not permanent at this stage. The other thing is that it actually, if you don't mind, Sal, scrolling down to the actual text right below that figure, because this is why I really think ENCODE was right to take a biochemical approach to this. I think their original conclusions got out over their skis and creationists glommed onto that. But right here, this explains biochemically why I think a lot of this stuff is non-functional, because they're saying, where do we see the greatest level of expression of these things? It's during this early stage of development, and it's 2 lines above where you have highlighted, where it says, notably during the global reprogramming of, it's in the second line, starting with notably towards the left, during the global reprogramming of DNA methylation and histone modification that takes place during pre-implantation and germline development. So what happens at this very early stage, because the cells are totipotent, is you are resetting the regulatory scheme. Now, as that embryo divides and the cells start to specialize, first into the germ layers and then into their specific tissues, as those signals cause those specializations to happen, you start getting persistent shutdowns in various parts of the genome, and they're going to be locked up and inactivated as densely packaged heterochromatin literally for the rest of the life of that individual. Now, that's all going to be cell type specific, right? As you said before, it's all very specifically controlled. The thing that's important here is that we get the highest level of expression of these elements. We get a burst of expression when we're resetting all of that stuff, when we're going from a situation pre-fertilization that's very tightly controlled to, okay, we're going to wipe the field clean and reset those regulatory schemes across the genome. That's where we get the burst of expression. That doesn't tell me that that stuff is regulating that process. It tells me you let it out of its cage for that very brief window in the life cycle. And then very shortly thereafter, following implantation in the endometrium, you shut all that stuff back down as the various cell lines start to develop and specialize. I think that's the most reasonable interpretation of that. And I can see the incentive for the people who did the study to say like, wow, look at this. We have these things that are mostly not expressed, but they're expressed very early in development. Maybe they have something to do with regulation. I think that's a fair thing to say, and that's definitely like pizzazz, right? That's the headline grabber. But I don't think that's the best interpretation of those findings.
Well, that may be the case, but what I was trying to point out when I made the comment that DNA is not a code, it is a chemical. It can be used to implement codes, but it's also structural scaffolding. And this whole thing with the 4D nucleome, the DNA can actually, it can be a scaffold for molecular machines, and also for the structure of the nucleus and how pieces of chromosomes interact with each other. I don't think we'd want to be cutting this out because it's going to alter the geometry of how the 4D nucleome is able to do regulation. So if this whole thing, again, we may have this discussion about the RNA, but it does have physical significance as a physical substrate, and that's where more I'm headed. I don't think we can minimize the importance of both the length and positioning roles that it plays, and also that it hosts lots of chromatin that can be modified and can also be utilized by molecular machines involved in gene regulation. So I think it's very early on. Not to mention the L1s have been indicated to be used in transposing in somatic cells in the brain. And that's another one I don't think I got to put in our debate because I may have run out of time.
Paper, yeah, that one I'm familiar with. And the thing with that paper is when you read that paper, the abstract makes it seem like one thing. And as scientists do, is you get that, you get that headline, right? You got to get that out there to get people on board. But if you go down and read, they actually say most of the changes that we observe in these brain tissues, neurological tissues, they're going to cause problems, and they're going to be selected. The vast majority are going to be selected against, which doesn't, again, causing a problem for something that is non-protein coding and that is mostly going to be methylated and packed as heterochromatin. Seeing changes to that, I think, does not indicate that that thing is functional. I think it very likely indicates the exact opposite.
And that's an open question. I read the same paper. I came away with a different impression and the other thing is every neuron is different. And it could be that this mosaicism, this difference in the slight differences in the DNA might be, it suggests to me that's one of the ways that these cells are able to differentiate from each other. And that's very important to be able to have neural circuits that know where information is stored to retrieve it, just like the way our memory works. So the other thing, just as an intuitive thing, this whole viewpoint, and I'm just going to zoom out a little bit here, just a little bit more philosophical. This whole viewpoint that evolution will create lots of junk, there are people that don't necessarily feel that way, especially the biophysicists, that the sentiment that I got at the NIH, it resonates with a lot of biophysicists. If it's there for a reason. It has a role because we see optimization on so many levels. Just generally the whole thing about saying this is junk, it's non-functional. I don't hear that from other quarters. They'll use words like it's elegant, it's sophisticated, it's crafty. And
so two things there. First, I actually have a specific question, if you know, with the 4D nucleome. So what we're talking about here, everybody, is where you have associations actually in the nucleus where you have different chromosomes that are held in close proximity to each other, because you'll have like a regulatory network where it'll be something on one chromosome and then a piece on another chromosome and then maybe a gene that's expressed on a third chromosome. And these different regulatory elements are held together in close proximity. So we tend to think of the DNA in the nucleus as just kind of jumbled up more or less at random. But it's not that. There are actually very specific associations that are going on. So my question, Sal, if you know, is what percentage of the genome is actually involved in something like that?
The 4D nucleome?
Yeah.
It's like the whole genome because I'll give a very specific example, like the
Well, sorry, just to clarify, I don't mean like, because chromosomes are held, like DNA is held in place. It's not just randomly floating. But in terms of like having specific associations where this specific region of chromosome 5 and this one on chromosome 8 and this one on chromosome 12, those things are always held together and they interact with each other. Like, how much is doing that kind of thing?
They don't have an answer right now, but the sentiment is because we're finding it on different cell types, it reorganizes depending on the cell type. So, I mean, there's a specific example that was kind of landmark, the FIRRE link RNA, where it has the chromosome, 2 locations on chromosome 2. The anchor was on chromosome X, and then there's chromosome 17, and it formed it like a transcription factory. And they said the implication, this factory doesn't exist in every cell type. Every cell type, what they're seeing is these topological, one thing that we do know between cell types, these topological domains and transcription factories, the profile is different between cell types. So that means the DNA, be it repeat or whatever, is being involved in being able to get these things in a 3D position based on time. That's why they call it 4D, because you have the 3 spatial coordinates and then the time coordinate. And I looked at this. I said, you know, that translocation would be really bad for some of this if there are translocations in the genome. So the synteny is also really important, but also for the non-coding DNA. So when I looked at that, I was just like, this is maybe as short an answer as I could give. All indications is a lot of it's involved. A lot of it's involved. And that's why I've been saying to the creationists, look at the 4D nucleome. This is, and I want you to look at DNA as not being a code. Look at it as being a scaffold. In fact, I highlighted here, they said, now this is RNA, but I'd like to say this is also true of the DNA. A lot of this acts as scaffolds. So these RNAs could also be acting as scaffolds. We just don't know. We're mapping it out. And
so here, the reason I ask about the percentages is because as I've been following the evolution creationism discourse, capital T, capital D, the discourse, since 2004, 2005-ish. And I've heard this before where, hey, look at this element in the genome that makes up X percentage of the genome. And some instances of that, we have biochemical evidence that it does a thing, whatever that thing is, right? It connects chromosomes across distances in the nucleus. It serves for ERVs with one example where, the promoters are still used, even so the ERVs are functional. And the reason I always want to ask about the percentages here is what ends up happening is you say, okay, we found some examples where an endogenous retroviral promoter is now used to regulate what are non-retroviral genes. They are human genes. Therefore, that is a functional ERV, it's functional endogenous retrovirus, and we should expect lots of functionality in endogenous retroviruses. ERVs make up about 8% of the human genome, and the vast majority of those sequences are super degraded. You get the terminal repeats on either side, but in terms of the promoters and the genes in between, there's sometimes there's sequence homology there, sometimes there isn't any detectable sequence homology, and sometimes it's actually been excised, it looks like. And it's just terminal repeat facing one way, terminal repeat facing the other way, and then where there should be all the retroviral stuff, it's all missing. But all of those things get lumped in under the category of ERVs. So when you look at first all of ERVs, and then you go, okay, but just the intact ones that still have, promoters that could do something. And then of those, you look at the fraction of that sequence that is actually promoters, you end up going from, up to 8% of the genome that's in this class, up to that 8% might be functional to, well, it's actually 1 to 2%. And then within that 1 to 2%, it's a tiny fraction of that is the actual relevant part. And it ends up at like 0.05%, right? Yeah. So I'm skeptical of these claims because I see the same thing happening over and over again with other classes of the genome.
Right? I would agree with you in that specific case, but I'm just throwing this out there that that I'm trying to be cautious about this, but on the other hand, I can't ignore the idea of this scaffolding for molecular machines, because I've seen enough examples, and this is a very hot topic in understanding gene regulation. Just this 4D reconfirmation based on cell type. This is
it's super cool.
I mean, this again goes in the question, what's functional? Okay, so you have a piece of lumber, and you could chip a piece away, and it's like, okay, it still works. It's not really, maybe we could dispense with this. Well, then you can just keep doing that until you dispense with the whole piece of lumber, or cut through it, and you're like, oh. And so
at some point you need enough structure there for it to work.
Right. And so, okay, so granted, one might make the argument that it could tolerate certain amounts of change. And then so it's like, okay, what piece of the lumber is functional? It's like, well, you could say it's all of it, but then you can delete parts of it, and it's, you can drill holes through it, and it looks still like it functions. So when we're talking about the DNA as being a physical scaffold, and again, this is why I want to get people away from thinking DNA is code. It's like I want you guys to think of it as a physical substrate,
as a physical molecule that has structural characteristics.
Right.
Yeah. Because
only 2%, 1 or 2% is coding anyway. You got to think, and then you have some of the break, and then of course there's part of the DNA that's regulatory. But then
this you go through sequence specificity, you run out a sequence real fast at like. So 3, 4, 5%, yeah. And then forget about sequence specificity after that.
Now, I do think I saw some biophysical papers, and I'm glad we're having this discussion, that the repetitive nature of it actually does help spontaneously organize the 4D nucleome. That you're getting the things to actually physically map together in the right way. And that's worth, and I was looking at this thing with this extrusion loops, and then also these CTCF binding sites. I just had to put all that.
I have no idea what those are.
This is really
no idea what those are.
Oh, okay. I had it in. I'm sorry.
Oh, it doesn't. Don't worry about it. It doesn't matter. It's just there's so much going. I don't know what most of the stuff is.
Yeah. No, I'll put some of this. This has been motivating. This is probably good motivation for me to put this. And besides, the boss said, maybe someday you'll collect all this together, and we'll just release a book on some of this, and most of it will be just more or less a review paper where we're trying to make it accessible. So we'll see. So I sort of have to do this anyway. But you've given me good input. I think your criticisms about interpreting the papers as not necessarily evidence of function, I have to review that. I really respect your opinion on that, because I do want to represent the papers accurately. So those are good criticisms.
I think we've been going for just a little under an hour, so I'm gonna just say kind of my final thoughts, and then if you wanna give your final thoughts and let everyone know what's coming up on your channel, if you have any stuff planned or anything. Sound good to you?
Well, I think. Yeah. Oh, yeah. I'm sorry. You wanted me to wrap up, or did you?
Well, I was saying we've been going for almost an hour, so I'll give just a minute or two of my kind of 30,000-foot takeaway. If you want to do the same, and then let everyone know if you've got stuff on the horizon that you want to alert people to. So the kind of two things from me is, one is a little more specific, which is, it could be a whole other hour discussion of like, what is function? Because here we are talking about ENCODE, and we actually didn't say what exactly, we said what isn't function, right? We said that ENCODE's original definition is probably too expansive, but we didn't say like, okay, so where do you draw that line? And what you just brought up with having lots of repeats and being able to lose some of them, but you have like a lower bound. So if you have 50 of something, right, and you can lose 10, but it doesn't matter which 10, are all 50 functional? Are 40 is that sequence 80% right? It's an open question of how you actually characterize function in that situation. And that could be a whole other, we could talk about that for 3 hours just on its own. So that's a whole other question. But the big takeaway that I get from this is kind of going back to what I said a minute ago about, so we have the regions that are coding and regulatory, and there's a few other specific things that are sequence specific that we're like, we know that that's functional stuff that's conserved across large phylogenetic groups. We have other things that are like structural, right? You've got telomeres and centromeres, and you've got spacer regions where between, you know, an enhancer and a promoter, you got to have a specific number of bases, but it doesn't matter what they say. So you get like the structural region where the size matters, right? And you've got regions that you might call associative, right? Like you're talking about where it's this thing matches up with this thing on this other chromosome. And that might be just biochemical, or it might be sequence specific. You put all that stuff together as far as what we've characterized, and not only do you top out probably somewhere between 15 to 20% of the genome if you're being generous, the bigger problem, I think, for a kind of aggressive interpretation of the data to show that says ultimately we're going to find that most of the genome is functional, is that we're running out of genome to characterize. Because while you pick up maybe a fraction of a percent with ERVs out of, you know, the promoters or something that end up getting used, you then write off the other almost 8% that's in that category. And you could do the same with lots of other categories. For every L1 sign or every L1 line we find that has a specific thing that we go, maybe that's preserved, maybe it does something, you've got a whole bunch of them that make up tens of thousands of bases in the middle of an intron that get cut out and degraded and aren't doing anything. So even if you, like, we keep finding these little bits that do interesting things that are possibly contributing physiologically in some way, but as we find each of those cool little bits, we end up eliminating a much larger portion of the genome from consideration each time, is what it seems like is happening. So at some point you start running out of things to add to that pile, and you top out. I think we're down to probably 15 to 20% of the genome that's like not super well characterized. Even if you put all that in, that only gets you up to like 30% functional, right? So that's where I come down on this, as like, in 1998, in 2005, and 2010, and 2012, there's a lot more that was not well understood. That in 2021, now we have a much better handle on this stuff. And much of it, as you and other creationists describe, much of it, surprise, it's functional, and it may be more important than we thought it was. But in finding that out, we also found out a whole lot of stuff that definitely isn't. And that's why I think at the end of the day, the functionality is going to come down at somewhere in the 20 to 30% range max, not in the 60 to 80% range. So here, go ahead, and don't forget at the end to plug your stuff.
Thank you. I'd like to thank Dr. Dan for this discussion. I solicit criticisms. I'm not here to win debates. In fact, I think trying to do science by debate is probably very ill-advised, because what ends up happening, someone will say something that's very ill-informed and they'll have to try to save face. That's not a good situation. And that's, I don't want to be, teaching the next generation to be trying to save face. If you're called out that you made a mistake, you got to acknowledge it. I mean, I worked in the aerospace industry. Anything that's safety conscious, if you're wrong, you better admit it, because people's lives and their welfare depend on it. And so these discussions, I welcome the criticisms. I think Dr. Dan and I agree about being a little cautious about the interpretation of some of these papers coming out. They're not easy to read. They are great to read if you want to learn something of how biology works. I'm not as hung up on putting figures on functionality. The genetic entropy models will succeed even if only 2, 3, 4% of the genome is considered functional, not in the evolutionary sense, but in terms of
talk about that.
Yeah. In terms of biochemical and physiological success, but so I'm not as hung up on that number. And the reason I mentioned the 4D nucleome, it's very hard to look at this. We cannot build anything of that sophistication that can change itself, reconform its 3D geometry the way it does in such an elegant way, where it's putting one segment of one chromosome in proximity to another segment in another chromosome. The amount of foresight that's needed to put this together is incredible. So it's just hard to look at the 4D nucleome and not think that this isn't being orchestrated and requiring a lot of the genome to be kind of just sequenced right, and also geometrically laid out on the genome in a certain way. So those are my thoughts. As far as tentative as I've been describing the issues surrounding ENCODE and 4D nucleome and functionality of the genome, I'm far more emphatic about the problem of abiogenesis. So some upcoming things that are happening is I will continue after shows related to James Tour's abiogenesis series. And I'm not as cautious in saying that I think the creationists have won that argument quite well. That I think the abiogenesis industry has had some issues that need to be cleared up. And so those are things that I'm going to be highlighting on my channel. I will not be as cautious in my wording on that topic as I have been tonight. But still, this is a great topic. And I'm glad Dr. Dan and he's given me a chance to kind of complete some of the stuff I was going to put on the table. And maybe given his response tonight, maybe it's just as well in my favor that we didn't talk about it, because I think you leveled a pretty good criticism tonight of some things I don't think I would have been able to counter in that short space of the debate. So my hat's off to you. And for that reason, the science students who are creationists think you're better than Ken Hovind. We would rather have you our teacher.
I appreciate that. Although, I mean, Ken Hovind, that's not a high bar to clear, really. But so Sal, thank you for coming on. I just want to say that I really appreciate this kind of forum rather than a debate kind of forum where we can really like get into some of the nitty gritty in a like you said, not having to save face kind of situation where we can say, oh, I don't have any idea what that thing you just said means. And like in a debate, you can't say that. But in a conversation like this, we can say that, and we can pause and explain it, and then keep going. I had a conversation, if anyone didn't see it, with Dr. James Carter on Monday. Same kind of thing, very informal, just talking about stuff and not debating. And that I thought was also really informative and useful. So I'm going to try to do more of this kind of stuff in the future.
Yeah, I mean, if we're, you're in an educational position, I'm somewhat in a different way. And so is Dr. Carter. We have to conduct ourselves differently than trying to do all this self-promotion. It's just not, for me personally, there's something ethically wrong if you don't try to give the other side a hearing and try to, say, you got to respect what this other person is saying. So
take it at face value, and then take it apart and look at it and see if it all works. That's what we're trying to do. So Sal, thank you so much for coming on. This was a pleasure, just nerding out about DNA for an hour, and like great way to spend an evening as far as I'm concerned. So everybody, I will link Sal's channel in the description. We're going to get a rebroadcast of this over there at some point. Sal, I'll also reach out to you via email. Just give me all the papers that you showed. Just give me a link so I can put that stuff in the description as well for people. But I'll catch you with email afterwards to get all that stuff. And as far as stuff I have coming up, everybody, I don't have anything scheduled right now, but I have a couple of things planned. So I've got a talk that I want to do about E. coli and genetic entropy, and the claim that I've heard relatively recently for the first time that bacteria would not be subject to genetic entropy for a number of reasons. So I'm going to talk about that at some point, maybe in the next few weeks. I also have a couple of book reviews. I got Rob Stadler's book here that I read and took notes on, but I haven't had time to sit down and like talk about it yet. So that'll be happening at some point. But so keep an eye out for that kind of stuff. My schedule is sporadic at best. So with that, I'm going to wrap it up. Thank you all for being here. Remember to, if you didn't like the video, share it, subscribe, do the same thing on Sal's channel. Let's get this over the 1,000 subscriber hump. Okay, everybody. All right. Have a great night, everybody. See you next time.
Take care.